white blood cell with the Hochschild dye, and that specifically stains a nucleus, which would stain white blood cells, and double-stranded DNA. And so we observed a sample of blood that was stained with this dye under the fluorescent microscope, and it works with a series of different excitation and emission lights. So this is the incoming light that, for this specific microscope that we use, reflects on the specimen at about 360 nanometers. And so the specimen absorbs the light and reflects the emission light at 460 nanometers, so at an excited state, which causes it to fluoresce, and that's what you see, is that this green light coming up, which is the emission light. Also, this dye requires a specific filter set, so if you used a different dye, you'd have to use a different filter set, and the filter set just consists of the emission filter, excitation filter, and dichroic mirror. This is what I observed while looking under the microscope, and this was an old blood sample, which might be why the white blood cells are clustered together, as you can see here. So this is just with white light, and then this is the image while it's fluoresced. And you can see the other red blood cells around the white blood cells. And also I worked on a red blood cell lysis procedure, which is just a red blood cell lysis buffer. So you add that to a whole blood sample and centrifuge and wash with PBS, and theoretically you should have a pellet of just white blood cells, but in this case we used an old blood sample from the volunteer donor, and we encountered some problems obtaining a pure white blood cell pellet. So using this equation, we tried to find what speed and what time would be the best to obtain strictly a white blood cell pellet. So by plugging in numbers, we determined that 300G or about 2000 RPM should be used. And I tried 4 different samples just to see about what time would be best. So I did 15 seconds, 30 seconds, 1 minute, and 2 minutes. Clearly there's a lack of pellet formed 30 seconds and below. And then 1 minute and 2 minutes, there's not much difference, so about somewhere between there you would obtain a pellet. However, the pellet that I got was red, showing that the red blood cell lysis buffer did not completely lyse all the red blood cells, so that could be either the buffer itself or just the sample is old, the blood sample is old. Counting cells can determine the amount of cells in 1 milliliter of sample, and you do that by multiplying the number that you counted in one of the squares by 10 to the 4th. And you can also use this equation. It's a really simple equation, and it'll tell you the concentration of cells in the original mixture. So this is really helpful for determining multiple different things, and makes that a little bit easier. This is just pictures of what it looks like under the microscope, and a zoomed in image. Another zoomed in. Another equation I used was how to convert between RPM and RCF, which is necessary when doing any kind of centrifugation procedure in the lab. And so you just plug in the numbers that you have, and you can plug in either RCF or RPM and get you one or the other.